Hello, BIO178, and welcome to our week 4 lab. So for lab this week, which is again in person, we're going to continue processing the samples that we've been working with throughout the past few weeks for our group projects. And so if you remember last week, what we did was extract the DNA from the tissue samples we prepared from the black turban snail, Tegula funnebralis, or red abalone, Haliotis rufescens samples. And what we're going to be doing this week is, first of all, quantifying the DNA that we extracted. So we did that whole process and protocol of extracting DNA. The first thing we're going to do this week is to quantify it, make sure we have DNA, see how much we have, and also see what the quality of that is by assessing a couple of different what are called purity ratios. So we're going to quantify it, and then what we're going to do once we've quantified the DNA is do polymerase chain reaction, or PCR, to specifically amplify the 16S gene of prokaryotes. So remember, we got our tissue samples originally from the turban snails and red abalone, but what we're really interested in is looking at the microbiome community of those samples. So our PCR, the primers we're going to use, are going to specifically bind to and target the 16S gene of all prokaryotic bacteria species. And so then we're going to make a bunch of copies of that 16S gene through the PCR, and then next week we'll check to see if that reaction was successful. It's important to make a bunch of copies through PCR. It's a way for us to check to see if that gene is present. And this step is also important because for the future sequencing, we want to make sure we have a lot of copies of that 16S gene of all the different bacterial species that are present. So in the next high throughput sequencing step, we can then get sequences back that represent each of those different bacterial species in the microbiome community. Like last week, it's important that you not only review this written protocol, but also take a look at the lab report as well, because I'm going to ask you, and it's important for us to jot down some of the different data we collect from this week's lab. So make sure you take a look at the lab report ahead of time so you know what you need to record while we're in lab. If you have any questions, let me know. Otherwise, I will see you soon.